Hello students, welcome back to the biology class. So in the last class we started the new chapter, the 13th chapter, that is photosynthesis in the higher plants. We learned that photosynthesis is the process of formation of organic compounds from carbon dioxide and hydrogen donor. Like it can be water, it can be H2S, we learned from the NCERT, using radiation or the light energy trapped by the chlorophyll pigment. So when we talk about chlorophyll pigment, we are talking about water as the source of the hydrogen, or you can say water is the suitable reducing power or oxidizable compound. Oxygen is evolved in this process because water is used as a hydrogen donor when we talk about the higher plants. And during photosynthesis, light energy is changed into chemical or bond energy present inside the organic compounds. And we learned the overall equation also of the photosynthesis. We learned that what exactly photoautotrophs are and how important is the ocean's contribution to the photosynthesis, and it is done by which two main organisms. We did the historical landmarks. We understood that what exactly Stephen Hales, who is the father of the plant physiology, contributed, what exactly Joseph Priestley with his bell jar experiment of keeping the plant and the mouse along with the candle inside, burning candle inside. And then he talked about that how Priestley from his experiment concluded that something is taken away from the air by burning or by breathing, and which is again contributed by the plant. And later how Lavoisier identified that oxygen is that gas which Priestley called as the dephlogiston, and then the carbon dioxide is the gas which Priestley called as the phlogiston. The names phlogiston and dephlogiston. Phlogiston is carbon dioxide, dephlogiston is the oxygen. We even understood the contribution of Jan Ingenhousz, that how sunlight is essential and how bubbles come, and those bubbles are basically the oxygen gas coming out. And then Engelmann's experiment of cladophora, or the action spectrum performed by the first scientist. So all these things we did in the last class, followed by the Warburg's experiment, Cornelius Van Niel experiment, Emerson and Arnold's experiment, Robert Hill, who demonstrated the photolysis of water, and he confirmed that Cornelius Van Niel's concept is correct, that oxygen comes out not from carbon dioxide but from the water. And this was again experimentally proved by Rubin, Hashid and Kamen, and we'll be learning about this in the class. Then the site of photosynthesis we covered in the last class, that the structure of the chloroplast, outer membrane, inner membrane, the gap between, and the two main things, stroma and grana, was covered in the last class. That stroma is the matrix which is rich in enzymes but does not carry any pigment and is involved in the dark reaction, and how grana, or the thylakoids or lamellae according to NCERT, at the site of the light reaction, is rich in the pigments but poor in the enzymes. And we learned that in the prokaryotic organisms they do not have chloroplast but they have thylakoids required for photosynthesis. We learned about the pigments which are involved in the process of photosynthesis, and these pigments are defined as substances which have the ability to absorb the light at specific wavelengths. And we did the structure of chlorophyll, and we understood even the universal photosynthetic pigment is the chlorophyll A. And every chlorophyll molecule generally will have a head and a tail. Exceptions are there, like chlorophyll C does not carry any tail. And then this head is made up of 4 pyrrole rings, and all these rings are attached to the central magnesium present at the center. And the phytol chain is a long chain of hydrocarbon. We learned the various the empirical formulas of various chlorophyll pigments as well as bacteriochlorophyll, and we learned that the chlorophyll A is bluish green in color, olive, and the chlorophyll B is olive color, and then it carries the methyl side group, and this one carries the aldehyde side group. And we learned that they all are soluble in the organic solvent. Now let's continue with the topic of the carotenoids. Now what are carotenoids? In the topic of 8th and 9th chapters, I have told you whenever the word OID is there in any biological word, at the end it will mean to say like structure. So carotene-like structure. So what exactly are the carotenoids? Carotenoids are yellow, brown, orange pigments. So remember, these will be the green shades of every pigment, and carotenoids are yellow, brown, orange pigments, and they absorb the light strongly in the blue violet region. So which region they absorb the light more? Blue and violet region. And now they are very important because not every piece of photon or packets of the energy which come from the sunlight will be harvested or picked up by the chlorophyll. So some energy or some wavelengths will be missed. So those wavelengths are picked up by this. This enhances the overall performance during the process of photosynthesis. So whatever wavelengths are missed by chlorophyll molecules will be picked up by the carotenoid molecules. That's why they enhance the photosynthesis. And another point, if chlorophylls are the only pigment to function, then they will be, if the chlorophylls are the only pigments to function, then they will be overburdened, and this overburden will cause them to break down, and these biomolecules won't function. So to take away their burden, carotenoids act like accessory pigments. Accessory pigments means something which are supposed to help in the process, overall process. And another thing is that they even help the plants against the process of photooxidation. Sorry for taking away the register from there. It was moving actually. So carotenoids prevent photooxidation of the chlorophyll pigments, and that's why they play an important role, and they are also the accessory pigments. Remember, even chlorophyll B is accessory, C is accessory, A is the only main pigment, and rest all pigments we are talking about in the higher plants, they are only in bacteria, higher plants, and these are also present in higher plants. So apart from A, leaving A apart, chlorophyll B, C, D and carotenoids, they all are the accessory pigment.